Okay, welcome back. So we're about halfway through chapter 8 now. You can describe to me what the central dogma is. You can describe how DNA is replicated, including a lot of enzymes involved in that. You can describe to me the process of transcription and translation. If I give you a piece of DNA, you could tell me what its complementary strand would be. You could tell me what the messenger RNA would be, and you could tell me what the protein would be. But now let's talk about mutations. So sometimes mutations occur. Everything on Earth, all life on Earth, evolved from one ancient parent organism, and we're all different species now because of mutations. So mutations are kind of important to our existence. Mutations can also be involved with genetic diseases. So sometimes all it takes is one little mutation to have a non-functional protein and end up with a disease. Mutations can also give rise to new species of bacteria. Recently we had a pandemic of COVID, which was a virus that evolved through mutations of other viruses. So a mutation is a change in the sequence of DNA. And when the DNA is changed, it will end up changing the RNA, which can end up creating a different protein. It can be neutral. So as we just saw, the degeneracy of the genetic code, some mutations won't end up with a different protein. A neutral change would also be if you have a different amino acid put in, but it's still able to fold into a functional protein, so you didn't lose function. Mutations could be beneficial, give a cell a new ability, give it a better ability to survive, to thrive in a new environment. They could be harmful and lead to a lost function. Some mutations are spontaneous. They occur in the absence of a mutagen, just things don't work perfectly sometimes. Some mutations are caused by a mutagen. A mutagen is something that causes a mutation. I would think of things like tobacco smoke, x-rays, sunshine. Certain foods or chemicals will have a warning that, this is known to cause cancer. That would indicate that is a mutagen in that bottle. The main type of mutations we're going to talk about are base substitutions. A base substitution or a point mutation is when you change one sequence in DNA. You just take one out, you put one in, or you change one for another one. So if you have an AT pair, you take out the A, you suddenly substitute it with a G, and then when that DNA is replicated, it'll be base paired G and C. So you have just mutated that DNA. There's three main types within these that we're going to talk about. One is a missense mutation. In a missense, you still have a sense codon, you still have something being made, but it's the wrong amino acid. So you have changed one amino acid. In this example, here is your DNA sequence that it should be, but here our AAA-CCG has now become AAA-TCG. That means in transcription, we have one different nucleotide in our RNA, and then one different protein. So that genetic code is going to read here to put glycine in the normal DNA, in the normal protein, suddenly a serine at the end of this protein. You've made the whole protein, but with one different amino acid. That one different amino acid can affect how the protein folds, so it may affect the ability to even make an active site. If it can fold into the right way, that one amino acid might make a difference in how that active site is able to function. So that one little difference can actually cause a disease. There are human diseases that actually are just one little nucleotide that changed one little amino acid that changed how the protein functions. The next type of mutation would be a nonsense mutation. So in a nonsense mutation, you change one nucleotide. Still here, this T is now being changed to an A, which means our RNA is different. And now, instead of making this whole protein, you have an early stop codon. So you don't make your whole protein. You make an incomplete protein. this is typically going to be pretty disastrous. You are very unlikely to have a protein that's able to function at this, depending on how quickly the stop codon was put in. Okay, so missense and nonsense. Those are the two types we've talked about. Then we have frameshift. Frameshift is when you put in or take out one base. So when you put in or take in one base, you are going to affect the codons for everything after that. So here it should be UUU as a codon, GGC, UAA. But by taking out this A, suddenly we have this is a codon, then this is the next codon. You've just shifted where it's reading, right? That's why it's called a frame shift. So instead of having all of the normal amino acids being put in, you have changed everything after that. So you've hugely changed a protein just by taking out or putting in one little base. There are also silent mutations or neutral mutations, which aren't going to affect the protein, mainly due to the degeneracy of the genetic code. And again, sometimes these mutations can be beneficial, giving a new or an enhanced activity of the cell. Sometimes they can be harmful by losing the ability of the cell to do something. The mutation rate is how likely it is that any random gene will mutate when the cell divides. The spontaneous mutation rate is around 1 in 10 to the 9th replicated base pairs, or 1 in 10 to the 6th replicated genes. So it doesn't happen very often. That's a 1 with 6 zeros after it. However, in the presence of a mutagen, that can increase to 1 in 1,000 replicated genes. We can use mutagens experimentally to create mutants, to intentionally create mutants, a mutant bacteria or mutant yeast or something for research to learn what is this gene doing or how does this affect the function of this cell. Actually, we'll come back and talk about how we research with that. Our cells do have the ability to repair DNA. So one example of DNA damage would be photo damage or light damage that can come in, and when there are two thymines next to it, it can trigger these two thymines to bind to each other instead of binding to their A's on the other strand. So photolyases are these light repair enzymes that they find these thymine dimers and then go through nucleotide excision repair. That means we excise or cut out the nucleotides, repair the mutation, and have healthy DNA. Okay, so how can a mutation be beneficial? How can a mutation be repaired? And how do mutagens affect the mutation rate? Okay, back to identifying mutagens. So we can use this test to learn about what a gene does, because by mutating a gene, it'll have a different function. We can also identify that something is able to mutate genes by doing this test. There's two different versions we can do. One is positive selection and one is negative selection. So positive selection is going to be able to identify a mutant because it grows when it shouldn't. So one example would be if you have your media and you add penicillin to it, your bacteria shouldn't grow because that penicillin should kill it. But those bacteria that have just gotten a mutation to be resistant to penicillin will grow. So that's positive selection. If something is growing there, you know it's mutated because suddenly it's able to do something new. Negative selection is when mutant cells are not able to grow. You end up with something that's called an auxotroph. An auxotroph is a mutant that has lost the ability to do something for itself. So many bacteria can make their own histidine. Histidine is an amino acid that's required to make proteins. If the bacteria is able to make it, it doesn't have to be fed it. But the auxotroph would no longer be able to make it. So it now has this new nutritional requirement. It has to be fed this amino acid. So here's an example of negative selection. I am growing these mutants on a plate that has histidine in it. So both normal cells and mutated cells are going to be able to grow. I'm going to stamp them on this little velvet stamper. And I have two new plates I'm going to grow them on. One is a media with histidine, so both mutants and normal cells will grow. And then one is a media without histidine. This will only grow the normal cells. Mutant cells won't grow. So here I can see which colonies are in both of them. And oh, look, this colony does not grow on the normal media. That means this is an auxotroph. It has been mutated where it has to be fed histidine. It can't grow on normal things. So that is an auxotroph. That is negative selection because it doesn't grow where it should. Okay, so be able to tell me what is positive and negative selection, or direct and indirect selection. Be able to tell me what an auxotroph is. This process of replica plating, this is how we detect where an auxotroph is. Carcinogens are mutations that cause cancer in animals, including humans. It's kind of important for us to know what is a carcinogen because people usually don't want to have cancer. So we can do what's called the Ames test to identify that a chemical can change your DNA. Basically goes through a modified version of the test that we just went through, replica plating, and seeing if bacteria are able to be mutated and then if they can be reverted. So a reversion would be a mutation that reverses the original mutation. So it's basically just a complicated version of what we just went through. I'm not going to ask you to explain to me how reversion works, but I want you to be able to tell me what is a carcinogen, and I want you to be able to tell me that the Ames test uses bacteria to test for something, if it is a carcinogen or not. Okay, so how could you isolate an antibiotic-resistant bacteria? How could you isolate an antibiotic-sensitive bacteria? What is the principle behind the Ames test? Okay, we still have a little bit more to go in this chapter. Some kind of microbiology-specific topics. We're going to talk about genetics. So come back and join me for our next chapter, or for our next discussion in Chapter 8. And I'll see you soon. Bye.